This presentation is about the summary of two papers related with axotomy, which means the neuronal axon transection. The first paper is laser-based single axon transection for high-content axon injury and regeneration studies, published in PLoS One on 2011. And second is axotomy depletes intracellular calcium stores in primary sensory neurons, published in Anesthesiology on 2009. In the first paper, researchers studied the retinal neuronal RGC5 cell line after laser transection for demonstrating that the ability of these cells to initiate a regenerative response correlates with axon length and cell motility after injury. They used the low-energy picosecond pulsed laser with 7 nanojoule pulse energy, 5-second exposure duration, 1064 nanometer wavelength, and 6 picosecond pulse width for axotomy. Firstly, they distinguished 3 groups of RGC5 cells based on morphological responses to axonal injury, whether they initially regenerated their transected axon. Group A is cells either resealed the injured axonal membrane and reversed axon transection, reconnected the transected sections, or grew new processes at the damaged site. Group B is cells that neither repaired nor replaced their injured axons, and Group C is injured cells died within 1 day of injury. There were 2 different features related with regeneration between these 3 groups: cell body motility and axon length. Results indicate that cells which regenerated their axons had a lower mean motility than cells that did not, and cells with longer axons were less likely to initiate a regenerative program after axotomy compared with those with shorter axons. Next, in the second paper, researchers examined the effect of injury on intracellular calcium ion stores of the endoplasmic reticulum, which critically regulate the calcium signal and neuronal function. Endoplasmic reticulum calcium ion stores released by the ryanodine receptor agonist caffeine decreased by 46% in axotomized small neurons. This effect persisted in calcium ion free bath solution, which removes the contribution of store-operated membrane calcium channels, and after blockade of the mitochondrial sarcoendoplasmic calcium ATPase and the plasma membrane calcium ATPase pathways. Calcium ion released by the sarcoendoplasmic calcium ATPase blocker thapsigargin and by the calcium ionophore ionomycin was also diminished by 25% and 41%. In contrast to control neurons, calcium ion stores in axotomized neurons were not expanded by neuronal activation, by potassium depolarization, and the proportionate rate of refilling by sarcoendoplasmic calcium ATPase was normal. Luminal calcium concentration was also reduced by 38% in axotomized neurons, in permeabilized neurons. So in summary, our first article indicates that RGC5 cell motility was related to the level of injury, and the liberation of chemoattractant molecules from the injury site depends on the injury level. And second article showed that the amount of calcium ion which can be released from stores through a variety of pathways is diminished after axotomy.